Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 88th seminar, and I call it University of British Columbia Day. I am attending the Synthetic Biology Gordon Research Conference at Sunday River, Maine, USA. I have met so many great people here, including many SynBYSS speakers and attendees from all over the world, and great to see you all. And if not this time, probably another place. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Nozomu Yachie. He is a professor at University of British Columbia, a Canada Research Chair, I believe one of the most honored title in Canada, and also an Allen Distinguished Investigator. I met him last year in person when I visited UBC for a seminar. We had such a great dinner at a dim sum place, which I still remember and miss. And of course, the best meal is what we have with great people. Also, great people and then enjoyable environment. So Nozomu, thank you so much for your contribution to the scientific community and education for future leaders. And also, I'm thank you because you're going to Japan soon, but you kind of share your time with us in this a little bit earlier time for your time right now. And virtual podium is all yours now. And thank you again. Thank you.
Yeah, not a problem. Thank you very much. Thank you for having me, Deslok. So yeah, I'm Nozomu Yachie from the University of British Columbia, and I actually recently started the satellite lab in Osaka University in Japan. So that's kind of the reason I'm doing back and forth these days. So my lab, I don't know how to introduce. Like we do lots of like a biotech development, and today I'll introduce one of the ideas that we have, which is a kind of crazy idea, but we want to have a video camera system within the cell to do new biology. And let me start like this. So, you know, like when we look at the animal body, it's beautiful, right? It's orchestrated with many numbers of cells, but we actually don't know how they behave at the cellular resolution or molecular resolution. And this is kind of a question that I want to tackle. So if I pick an example of development, you know, our body starts from single fertilized egg, and this divides into 2, 4, 8, and so on. And eventually we get animal body, or if you look at my body or your body, it's super complex, and there are like you know, billions to trillions of different types of cells. It's a beautiful process, but the problem in developmental biology is that, you know, if you want to observe tissue section of this mouse, you have to kill it. Well, these days we can do single cell transcriptome or single cell genomics, but in order to observe them, you have to break them. So unfortunately, in any area of biology, when we want to do observation, we have to destroy them. Therefore, we can do observation only at the time of observation, and we never know the dynamics of them, or we never know how they will behave if we don't sacrifice them for observation. So in order to tackle this issue, and I'm pretty sure there are many people working on this field these days, but we kind of have an idea of DNA event recording. The basic idea is we want to put the video camera system to the cell. Of course, it's not going to be a plastic video camera. It will be made of protein, RNA, and DNA. It's a super miniaturized camera system within the cell. And let's imagine that such a small device can do observation what is going on inside a cell, outside a cell, and record you know, information into a hard drive. but of course we don't have a hard drive. But you can remember ATZ cDNA sequence is a digital information storage media. So we use synthetic DNA to record event information there. And let's imagine that we can have such a cell having video camera system and let develop, for example, mouse system. And at the time of observation, we of course need to sacrifice them, unfortunately. But when we analyze them at the single cell resolution, these days we can read out the DNA memory from each single cell. So we kind of envision that we can reconstruct what was going on in the past computationally in a large scale. Sounds like sci-fi, I know. And saying this sounds easy, maybe, but it's actually super tough. So we kind of identified 4 different technology pillars with the analogy of the video camera system. We got to work on developing synthetic memory system within an animal body. So my lab is hardly working on mouse engineering and thinking of how to put lots of synthetic DNA to mouse chromosome. And the second pillar is like a lineage tracing, or we want to sometimes sense like a gene expression patterns. But once we sense these event information, we need to transmit that to the memory system. So we need to develop a writer system. And this is kind of the why my lab's history has been on the genome editing field. But the most important part is once you record the memory to the DNA tape, you have to read it out by sequencing, and you need to reconstruct, the past event information from the sequence of DNA ATGC. So we got to think of high performance computing technology, as well as like a new kind of, phylogeny estimation algorithms, or, the in general, like event reconstruction algorithm. So yeah, today I'm not going to introduce any of them detail, but this is like what my lab is doing. And the next one is my last slide, but the most important one for me. So I don't know, like I self-identify I'm a crazy guy, and I always say stupid stuff in the lab. I have lots of like talented people in my lab, which I am proud of the most. They sometimes get mad because I'm saying too crazy stuff, but they picks up idea. And I have people who does mouse engineering, computational biology, synthetic circuit generation, and genome editing, and they are all independent and working together within the lab. So they kind of always like collaborate within the lab and make a big impact, which I appreciate. So I'm proud of that. My lab is super interdisciplinary. But at the same time, even my lab is interdisciplinary. Like what we are doing is hard task. So we need lots of other collaborators internationally, and we are also super fortunate by being surrounded by these people, which I cannot name all of them. But if I would deliver one message to everyone. Thinking big is fun. Taking risk is fun. Being fearless is fun. And I have never seen who failed by being fearless and taking risk. Probably they are because they are not, survived, and that's why I don't see them. But I believe that taking risk is the best strategy, and thinking big is fun. You should, we should all pioneer the field and change the world. So yeah, that's it. Thank you.
Absolutely brilliant talk. I mean, I 100% agree with you. I mean, thinking big, dream high, and there are too many global problems we have, but I guess, we have also passionate, enthusiastic, student and postdoc wanting to solve those kind of problems. And I believe by encouraging, inspiring those people, we will solve those problems together. And then you give absolutely amazing vision to us. And then your perspective paper, I believe I read that paper, and that was absolutely brilliant. So thank you so much. And then I absolutely honored to have you today. Thank you.
Thank you.
Okay, so now the main speaker of today with a longer introduction. Ania Baetica is an assistant professor at Drexel University in mechanical engineering and mechanics. She leveraged control theory along with synthetic biology, systems biology, and computational science to solve biotechnological and medical challenges. Her research is on the design of robust and modular plug-and-play synthetic circuits with biological feedback. She obtained her PhD in control and dynamical systems from Caltech under the guidance of Professor Richard Murray, and her bachelor in mathematics from Princeton University. She also worked as a postdoc at Professor Hannah Al-Samad lab in UCSF. Actually, the Hannah actually gave a pioneer talk a couple of months ago, I believe. I would say that she is a true expert in control theory, mathematics, and computational biology. And then she is a really, really real rising star with a huge potential. So Ania, thanks so much for your time today. And please take it away. It's all yours.
Thank you, Taesuk. Thank you for having me and inviting me and creating this seminar series. It's very nice of you, particularly during the COVID pandemic when we weren't sure that we'd be able to give conference talks anymore. Today I want to talk to all of you. As Taesuk mentioned, my name is Ania Baetica, and I'm an assistant professor at Drexel University. Even though I'm in mechanical engineering and mechanics, I do the computational and theoretical work for a lot of synthetic and systems biology. So today I want to talk to you about my work, which I've titled Computational Pipeline for the Prediction of Modular Synthetic Circuits. But first, I like to give a little bit of an introduction about systems biology and synthetic biology, because they're both kind of newer branches of biology that are less traditional than, let's say, developmental biology that was mentioned a little bit earlier. So in the past couple of decades, synthetic biology in particular has had a huge impact and huge potential across a variety of fields. For example, in industrial biotechnology, synthetic biology can be very helpful at scaling up industrial processes. Similarly, they've both had tremendous potential and also tremendous deliverables in health and medicine, in the treatment of infectious and non-communicable diseases, as well as in things like cancer treatments and healthy aging and microbiome therapies. And if you want to learn more about synthetic biology, I think the roadmap from EBRC, the Engineering Biology Consortium, is a great place to start. Let's dive a little bit more into how to think about synthetic and systems biology. I think of them as two different frameworks that can help for prediction and engineering design without having to keep track of all of the details. And in particular, I like to think, this is just my perspective, but I like to think synthetic biology is analogous to engineering and systems biology is analogous to physics. And the reason for that is I think that synthetic biology basically kind of abstracts away details and allows you to build things that are complicated without having to understand all the details of every single part. So I think of it as analogous to engineering, where you can build a laptop without having to necessarily know all of the details of every single microprocessor. And I think of systems biology as also an abstractionist framework that relies on things like emergent properties. So you can think of this, it's analogous to physics, where you don't need to know about every single gas molecule inside a hot air balloon, but instead simply knowing the emergent properties of heat is sufficient to understand how this balloon rises. So both synthetic and systems biology are frameworks that rely on abstraction, and they do prediction and engineering design without having to keep track of all the details, allowing us to make faster progress this way. So from my perspective, predictive models are actually key to being able to make this level of abstraction. They're key to understanding and engineering complex systems that have multiple components. And I think a really simple example of it that I like to give people is that if you start off with a little circuit inside the cell that's made up of 3 genes, and gene A is repressing gene B, and gene B is repressing gene C, and then gene C repressing gene A, then if you look at the protein levels, because gene A is repressing gene B, when it's high, the blue of gene B will be low. And then gene B represses gene C, which means that the yellow of gene C will be low, and so on and so forth. And this creates a very nice oscillator. However, if you just simply add one more gene to the system, the oscillation disappears altogether in this 4-node system. So to be able to keep track of and understand this kind of property and how this design results in these kinds of properties, I think you need predictive models to tell you that if you change the system, how will it behave? And in particular, the systems biology and synthetic biology perspectives, I like to think of them as bottom up and top down a little bit. So the systems biology perspective is that you do analysis through predictable models of the layers of abstraction below. So when you move from genes to biomolecules to cells to organs, you do some abstraction, and then you have predictable models that tell you how these abstracted layers below behave. However, synthetic biology perspective is that you perform engineering design through modularity and robustness to the layers below. So like I was referring, you can think of it as trying to build the laptop, but without having to keep track of all of the details of the processors inside of it. So for my own research, I like to think about the question of how can we do design that's robust with respect to lack of information in the layers below. So we're never going to know everything about all the biochemistry of everything. And even if we did, it would be utterly overwhelming. So then I like to use these abstractionist frameworks of systems and synthetic biology to ask the question of how can we do design that's robust with respect to this lack of information. And there are several obstacles to performing biomolecular system prediction and design out of multiple components. They're well known in the literature. One of them is that these biological circuits are imperfectly modular. So when you combine 2 of them together, their properties can change in really unexpected ways. Another drawback and challenge in the literature is that, as I've mentioned, there's incomplete characterization of biomolecules and their interactions. We have unmeasured biochemical kinetics. We have unmodeled higher order dynamics. And here I have, as an example, the toxin and antitoxin system from E. coli, CCDA, CCDB, where there's quite a bit that we don't know about the biochemistry of it. So to try to address these challenges of imperfect modularity and lack of robustness and lack of knowledge about biochemistry, I personally like to come up with a couple of different solutions. So one of them is that I give engineering guidelines of how to design these biomolecules. I like to use modeling with uncertainty to improve the accuracy of our predictions. And I also like to use biological feedback to mitigate against this lack of robustness, as I call it. So in this particular project, I'm interested in how to modularly compose synthetic circuits that have unmodeled dynamics and imperfect knowledge of the underlying biochemistry of the system. So in my postdoc work, what me and a collaborator of mine did were that we developed these 8 composed synthetic circuits. And what's really cool about them is that they're modular. We start off with a basic circuit, and then we just keep adding pieces, more and more pieces to it. And we're able to show that we actually have modularity, which is very rare for synthetic biological circuits. This is a property that often fails in practice. And it's required a lot of care and thoughtful design to kind of achieve this. So to get a little bit more into the type of circuits that we have built, we were interested in pulse generators. So pulse generators create a pulse of gene expression, and ideally, they respond to something like an external input by creating this pulse of gene expression and then returning to the same level over time. And this property is known as homeostasis, or sometimes perfect adaptation. And the reason why we were interested in building these pulse generators is because we were hoping to do targeted drug delivery. As I mentioned, synthetic biology has a huge amount of promise for health and medicine. And in particular, we were interested in putting these pulse generators inside cells, and they could do a little bit of therapeutic release. So in this blue cell right here, they could deliver, let's say, a drug, but it would be a controlled radius of action as represented by the orange cells, because this pulse of therapeutics would go up and then go back down. And that way it wouldn't spread to everything all around it. So we were hoping to synthetically engineer cells with this kind of pulse generator that could deliver drugs. So we started off with a really simple design, and actually what we observed was that we had a lot of challenges even in the simplest version of this. So in particular, the first design for a pulse generator that we made, it had about 30% error. So as I mentioned, we were hoping for a homeostatic response. That means that the pulse starts off and then comes back to the exact same level. But instead, we observed the error here, and it was quite large. The other thing that we had a challenge with was the amplitude of the pulse. So we want a pulse that's pretty high, you know. And what we observed was that we could get about 6 times over the basal. So the basal is kind of where we're starting off. So our question when we encountered this was that this pulse generator was not homeostatic, and we were wondering how can we go about fixing it. So we know from engineering, which is kind of my area of expertise that I try to apply to synthetic and systems biology, that negative feedback is known to restore homeostasis. So we have negative feedback in a lot of electronic and mechanical systems. So for example, when we're driving in a car and we're going up the hill, negative feedback can account for things like not really knowing how many people are inside the car. So it can deal with this kind of uncertainty, which is very commonly found in biology. So we thought that maybe if we add some negative feedback to this pulse generator that we were struggling to obtain homeostasis for, that maybe that would be sufficient to fix it. And spoiler alert, this was correct, and we were actually able to improve this pulse generator using models with uncertainty quantification to kind of predict what adding negative and then subsequently positive feedback might do to this pulse generator. So we started with the pulse generator design, and we tweaked that by adding biological feedback in a modular way. So this is a really novel idea. Most of the time when people want to tune the behavior of a synthetic biological circuit, they just kind of swap out promoters and parts of that circuit. However, what we did was that we used feedback to tune the behavior of it. And actually, you can prove that unfortunately just swapping out parts for the circuit would not improve the homeostatic result that we got. Yeah, so to get a little bit more into the details of like what this pulse generator was and how we built it. So this pulse generator, for those who are interested in the biological details, we actually built this pulse generator in yeast to begin with. So the way that it works is that it's a network made up of 4 genes, GEM, SynTF, key, and cage, and it responds to induction from the hormone estradiol. So when estradiol is added to the circuit, there's an inducible transcription factor that responds, and it creates a pulse of gene expression. I want to focus in a little bit on the system of the key and the cage. So this is something called Degron-Locker. It was published by the Baker Lab, I think, in 2019, and we just co-opted it with their approval for the circuit. It's a de novo protein degradation switch, and the way that it works is that there's this key molecule that when it's exposed to the cage molecule, it opens up the cage and the key binds, and it uncovers this degron molecule, and this degron is exposed, and then the complex is marked for degradation by the yeast proteasome. If I correctly recall, the Baker Lab as well as the Elsamont Lab were working on a mammalian version of this. The challenge of working with the system of a de novo protein degradation switch is that it hasn't been measured in vivo. It's a complex protein-protein interaction that we don't fully understand. So this is where modeling and engineering come in. We can do a bunch of experiments to get to all of the details of how this works, or we can try to live with the uncertainty of how the system actually works. We know a little bit about it, but we don't know everything about it. So as I've just mentioned, when you add hormone estradiol to this circuit, what occurs is that first the cage is activated, and this makes the pulse go up, and then indirectly the cage is inactivated through the second arm of repression of the circuit through the key, and then that's when the pulse goes down. So basically, the pulse of gene expression is created through the direct activation and then indirect and delayed repression of the cage molecule. And we're able to measure this using YFP, yellow fluorescent protein that's fused to the cage molecule. So as I've just mentioned in the previous slide, due to the fact that we have uncertainty in how things work, and due to the fact that we don't really understand why this is not a homeostatic circuit, we need to come up with some mathematical model for it to try to represent the experimental data that my collaborator has measured. So in particular, I developed a mathematical model of ordinary differential equations of this pulse generator circuit, and I fit it to the experimental data that we had using Bayesian inference, so that I can account for this uncertainty in the biochemistry and in the fact that the model is a poor representation of reality. All models are poor representations of reality, more or less. Okay, so here, using Bayesian inference, the only thing that I want to show is that in the dots, we have the data, and in orange, we have the mathematical model fit to this data. And you can see, of course, that there is some error because the model is not perfect. However, it is good enough to proceed with trying to make a prediction of what might happen if we try to add negative feedback to try to fix the system and its property of homeostasis. So after we do this fitting, we can mathematically predict what adding negative feedback does. So this can precede an experiment that confirms the same result. So in particular, here I have the prediction of what adding negative feedback to the pulse generator might do. I've split it up into 4 quadrants. Each quadrant represents whether the amplitude of the pulse improves or worsens, and the same quantification for the error, or in other words, for the lack of homeostasis. So the 4 quadrants are better amplitude, better error, worse amplitude, better error, worse amplitude, worse error, and better amplitude, worse error. And we're trying to see where this circuit will fall upon adding negative feedback. And we can see that in about 46% of cases, we'll get better amplitude and better error. And in about 36% of cases, we'll get better amplitude but worse error, and so on. However, there is one of these quadrants that are actually infeasible biologically, because they correspond to the feedback being broken. So we can actually eliminate one of these quadrants because it's not biologically realistic in our prediction. And what we find is that there's a 46% chance that we get better amplitude and better error, and 17% chance that we get worse amplitude and better error. And where our experimental circuit that validates this prediction falls is in this particular quadrant, the 17% one. So subsequently, we may want to improve the pulse amplitude. And one good way of doing that also comes from engineering design is that we may want to add positive feedback to the circuit. So we're hoping that positive feedback improves amplitude, because this is our general understanding from engineering. And here I've made a similar prediction where I've split it up into 4 quadrants. And here you can see that 78% of circuits fall into the better amplitude, worse error quadrant. And when we build this circuit with positive, the pulse generator with positive feedback experimentally, it in fact falls into this quadrant of 77.5% prediction. Once we build these circuits experimentally, we can collect data from them, and we can go back and calibrate these models to be even better than what we had before. So here you can see that when I fit the mathematical model of the pulse generator with negative feedback to the experimental data, I get a pretty good result. And the same happens for the positive feedback. So we just go back and forth between doing modeling to try to understand what the experimental circuit is doing. And then we make a prediction of what adding feedback to it might do. And then we calibrate those models. So by going back and forth like this, we can actually do this framework of modular synthetic composition for 8 circuits. We start with the pulse generator design, and then we improve on it. We go back and forth by tuning its response with positive and negative feedback. And here we've also built a collection of filters to go with our pulse generators, and we can also tune their behaviors with positive and negative feedback. Subsequent to that, we perform these quantitative fittings to calibrate our models as we go along. And ultimately, we are able to reproduce experimental data, and we are able to live with the fact that we don't know everything about the biochemistry of how these circuits work. So in this project, I use biological feedback to mitigate against a lack of robustness, and to improve the modularity of these highly composed synthetic circuits. And I also used modeling with uncertainty to improve our lack of robustness to this lack of knowledge of biochemistry and certainly unmodeled biochemical dynamics that are so inevitable in synthetic biology. Okay, beyond this project, I wanted to introduce my own research group at Drexel. I'm a new PI. I worked at Drexel for I think 4 months now, so I'm really brand new. What I'm hoping that my lab will do is that we'll use biological feedback design to address biotechnological and medical challenges. And in particular, I have approaches coming from synthetic biology, systems biology, and engineering and computational science to try to solve a variety of problems. So something that's of interest to me is trying to understand what happens when you add multiple biological feedbacks and how you can use this to better engineer devices that are put inside cells. Another project and research direction that's of interest to me is that I would like to improve the targeting of drug therapeutics by understanding how multiple feedbacks work together in these conserved signaling pathways like MAPK and RAS to develop drug resistance. So hopefully this will have an impact on improving drug therapeutics for cancer treatments. And then lastly, I'm interested in microbiome population control. So that means composing populations of microbes without letting one of them take over the entire population, but instead having really tight control over how they interact with each other. And hopefully the application area is microbiome therapies. I'm currently still hiring students, graduate students or postdocs. This is my postdoc NAM, and I have an incoming graduate student. I also have a summer student and a co-op student. So if you're interested in this type of research, please feel free to reach out to me and join my research group. You should find me on the Drexel website. And with that, I want to once again thank TASOC for inviting me, and I'll stop here.
Oh, wonderful. Thank you so much. It's absolutely amazing. You know, you are really genius in mathematics and control theory. I now learn a lot. So Nozomu, do you have any question or comment?
I do. Actually, I have a very naive question to the field. So DNA is only single copy if it's bacteria. If it's diploid, it's 2 copy. And in general, within single cell, the number of molecules or players are very limited, right? And
Yeah.
but I believe that you're simulating this as a population average. And my question is population average simulation actually recapitulate the average of these noisy single cell behaviors?
I mean, that's always a valid question for every time someone is using population averages of everything. So the data here was taken with flow cytometry. Here, let me pull up some of the data. This is flow cytometry, and as you just guessed, it's across the population. You can definitely model everything stochastically using things like the chemical master equation, chemical Langevin equation, and so on. The problem with doing that is that, well, not the problem. Sometimes it's very useful and very informative, but the drawback of doing it is that the models become a lot more complex very quickly. So for something that has 4 genes like this, the model would be a little bit large. But yes, it's always an option to try and see if it makes a difference or not.
I see. But is it usually practically, okay? Like if you simulate single cell separately and take population average, do they usually become similar to the population average models?
Sometimes, and sometimes they behave really interestingly. So there's this series of papers that says that biology actually makes use of this variation inside cells, inside single cells, to achieve like really important functionalities. So are you in one of those cases, or are you not? You know, that's always a really hard question to answer. Yeah.
I see. I was also wondering if you could just generate random circuits first, like by like random DNA assembly, and fact sort this population with the certain GFP threshold, and retrospectively identify circuits or something like that.
Yeah, I think probably you could generate some kind of like library of circuits. I think here we were going for some pretty specific functionality that we wanted it to do, and we're also going for understanding how it achieves that functionality. When you do like a random library screening, it's usually like you make like so many circuits, right? I'm not sure how you would build a library in this particular case. I guess kind of just keep adding genes to it. I'd have to think more about it, but yeah.
And how big can you go? Like, how many components can you control? I mean, like, you know, you talked about uncertainty, but and this is at the moment on this slide you show us like a 4 component system, and later you showed us like 6 or 7 component systems.
Yeah.
How big can we go in you know, microbacteria probably?
Yeah, so like this one with 1, 2, 3, 4, 5, 6, well, there might be other stuff in it as well. Actually, the size for synthetic biology is very small for the circuits that we have nowadays. Building something that's even like this big and having it be modular, and what that means is like you know, if I remove this part of it, the progesterone arm of it, it still works as I would expect it to work. This is very hard. Is my general understanding. Like all these circuits are functional within just by themselves, and then you keep adding feedback on top of them, and that's facilitated by that de novo protein, because you can just put it on anything. You just keep adding and adding and adding feedback on top of it. That's actually really hard to do. Like I can't really think of a lot of papers out there where they tune the behavior with feedback and they compose multiple feedbacks on top of each other. I think it's just us and maybe like one other paper that has ever shown that experimentally. So yeah, unfortunately, this is kind of the state of the field. Now your question is, you know, can we just keep going from this one? The answer is probably a little bit more. I'm not sure. We'd have to do the experiment, but yeah. Synthetic biological circuits have remained fairly small in size, I would say. I think maybe Tae-seok has more to say about that.
You know what, I claim 2012 in my Nature paper, we construct and build the largest circuit in the world with the 4-input AND gate. And since that, Chris Boyd, my advisor, also put a lot of effort and then published one paper in Science with the gigantic parts, and then building everything using Cello, the you know, computational algorithm. But the size-wise, I mean, also number of parts, I mean, similar to my manually constructed, you know, circuit. So we don't know what is the limit, but to me, I mean, most of the applications do not need 4-input AND gate or 4-input circuit. I think most of the circuit probably require just simple, even 1-input circuit. And then because, I mean, if your circuit become too big, I mean, there are a lot of problems, including retroactivity, burdens, whatever. And that potentially lead to mutation, accumulation, blah, blah, blah. So fundamentally, the bottom up is very important. because we need to understand using bottom-up approach, and then also systems biology giving some another insight, but bottom-up is very important aspect to understand life and system. But in terms of real-term application, I believe the simpler the better. I mean, in the future, with a lot of technology developed, we may have very complex circuit that might be better, but that's my personal opinion, but somebody else probably thinking differently, but that's my personal opinion about complex circuit. Because I believe in complex circuit, because that's why I built a complex circuit, but I changed my perspective right nowadays because I'm more going toward practical application, and then I realized complex not necessarily good. I mean, for real world application.
Yeah, I think the question is, what do you need a super complex circuit for, and can you do it with something that's simpler as well?
Right.
I think a key selling point of this work is that actually we don't have the problems that Tae-Suk mentioned with retroactivity and
Right.
all sorts of other stuff, and it's because we are using a de novo system with a Degrun locker. If we did use something else, we would run into a lot of problems with what the cell wants to do, because it wouldn't be orthogonal to everything that's happening inside the cell.
Right. So we still have very simpler circuit compared to the entire whatever, even if simple E. coli entire genetic network. Our synthetic circuit is very, very, very small because we are talking about only handful genes, and then reporter, and then regulator, and so on. So compared to the huge network, I mean, we still limited.
Yeah.
So we need to do that.
Orthogonality is the key to make it more stable. Is that what you're saying?
Can you repeat that again? Sorry.
Is that orthogonality the key to make it more scalable? Because like here you show bandpass filter, and you also show positive and negative feedback, which I assume make the output more stable. And you guys have all of these tricks now, and I was wondering what is the bottleneck to be scalable, and is that orthogonality to the native biological system, or where's the hurdle here?
Yeah, so the reason why we can even do this at all is because of the key cage system that is de novo, and therefore orthogonal to the things that are inside the cell. So we can keep, we can add it on various proteins, and then we can keep, like you see right here, it's added in one place, and right here it's added in another place. So we can just keep attaching it to different places to try to tune the behavior of these circuits. I mean, I guess you could attach it indefinitely. At some point, I guess my question is, what is the benefit and functionality that you are looking for? You could try putting some here and here and here and everywhere, right? Is that something that you're trying to do for a particular purpose? Like maybe the point that is not coming across is that we are looking for specific ways of tuning these outputs. Like the reason to add negative feedback to it is to improve homeostasis. So I can easily articulate the benefit of adding negative feedback here. For positive feedback, it improves amplitude. So I can easily articulate the benefit of that. If you keep adding it just for the purpose of scaling things up, I guess my question would be, what kind of benefit are you hoping to get outside of just having an increasingly bloated circuit?
Well, I want to make a video camera system within the cell. So that's the
Okay.
Yeah.
So my comment on this complexity scaling up. So the one challenge we have is not necessarily the only challenge, but also one challenge is we only rely on, so far, most of people rely on GFP reporter to analyze circuit. The problem is GFP signal is good, but most of the case we need multi-copy plasmid because of that convenience. I mean, if you integrate everything in the genome, that's one copy, and then typically GFP signal is very weak. Because of that, one problem we currently having is if you have the one negative feedback, that should be okay. But if you have multiple feedback, every single time you add one more regulator, that means multiple copy of regulator, because the copy number is, a lot, at least 5 or 10, whatever. That means you have now basically trade-off adding regulatory element that stabilize or make the robust system, but at the same time, the additional regulator become a burden. So that trade-off, you need to have very fine balance, but that is not easy task to do. So that's why a lot of people rely on computational, analysis, kind of fine-tune, but it's still, this is kind of very challenging job to do. So that's kind of my perspective, because bacteria evolved to have many regulatory architecture with including positive feedback, negative feedback, but we are talking about only one copy system. But now we have additional burden. Bacteria not necessarily require those kind of complex genetic circuit, because this additional circuit we impose for their survival, they don't need actually. So they tend to remove. So the most important thing is mutational stability of those circuits. And then that is kind of one complexity, one challenging, I believe, we have in terms of scaling out. That is actually my answer.
Yeah, I think we're not even getting into the mutational stability. It's very hard. That part is very hard.
Right, exactly.
Yeah.
Do you have any other question or comment?
No, thank you.
Okay, so I want to address the audience question from Maya. The first question is related to Nozomu's question. Library that screen for circuit topology or circuit parts. I forgot what this means.
So it's referring to the suggestion that we just screen for random parts that I think try to achieve the same functionality, if I understood the question correctly.
Okay. And then another question is, how do you design for more control without adding more load? RNA device can relieve burden, but do you work fast? Do they work fast enough for precise control or not really?
They can, and maybe going in that direction is another way of doing control. Here, I think you can do it through RNA devices, or you can do it the way that we have done it.
Okay, and then the last question is, what are your thoughts on orthogonal parts and building new proteins like Degron locker to replace the native parts?
I think they're great. I'm very excited about all the proteins, the de novo systems that are being created. The problem with them is exactly the thing in this talk, which is that because they're created at a really fast rate and published very quickly, they're not always fully characterized. So we need to live with the uncertainty that they introduce. So I love this idea of building de novo systems. I think it can give us the option to not run into some of the issues that were mentioned in this talk when we're building synthetic circuits. But oftentimes they come with their own challenges. They're not fully characterized. We don't fully understand how they work. And I think that that's okay. And I think we should capture that with modeling. And I think we can still get great functionality out of them. So yeah, I think this Degron locker system was super helpful in doing all of this work. And overall, I'm a fan. Yeah.
Wonderful. And then we did great job also in terms of time. I guess if you, I don't see any more questions. So let's close. So thank you so much all for joining and staying today. We are basically competing with the Synthetic Biology Golden Conference, and many of the regular attendees for this seminar in the conference right now, listening to Harris Wang's talk, I guess. So we'll meet again next week on July 27, Thursday, the same time, the same Zoom link. We'll have Professor Wilson Wang at Boston University and Ice Kiati Siwi at University of Washington. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stopped recording. Thank you. Give me 1 second.